I realized I never explained how I got my arm back. Because you guys might remember a couple weeks ago, Vader cut it off. And, it's quite a simple explanation. I just fired up the cloning machine and grew myself a new arm. Really not that complicated. So today, I'm going to talk about how prokaryotic cells evolved into eukaryotic cells. Now, before I get into how a prokaryotic cell evolved into a eukaryotic cell, I should probably explain what each of these cells are. And I know I did a video about this a couple years ago, and I'll put a link to that in the video itself. But I'm also going to do an update here because it's important information and I want you to get that information. And here is our prokaryotic cell with the black being the cell membrane, which is a kind of flimsy, not very protective cell coating. It basically keeps the cell goo and liquids or whatever inside the cell. And then the blue is the DNA, which you probably know what that does, but it's the instructions for the cell that allows it to replicate itself. The green is organelles, which in the prokaryotic cell's case is just ribosomes, which make proteins. And the red is the cell wall. Normally, it's not that far out from the cell membrane. It's actually, right around it, in close contact with it. But I'm not a very good drawer, so whatever. But the cell wall acts as more like the structure for the cell, because the cell membrane isn't very structured. Now we have the eukaryotic cell, which is much bigger, close to 10,000 times in volume. So, just a bit bigger. And whereas prokaryotes are used to make bacteria, and like archaebacteria, eukaryotes make animals, plants, fungus, and also protists, if you've heard of those. And then in black, we have our cell membrane again, with this inner black circle being the nucleus, and the blue again being DNA. And then our green dots are ribosomes, which again produce protein. And then here we have other organelles, which I'm drawing poorly. The mitochondria, the Golgi apparatus, it's kind of what it looks like, the lysome, and then the endoplasmic reticulum, that long squiggly one. The mitochondria takes oxygen and converts it to ATP, which is used as energy, has a lot of energy. The Golgi apparatus packages and sends out protein. The lysome breaks down and digests food. And this, the endoplasmic reticulum works as the highway in the cell, allows for fast transport. And the eukaryotic cell actually does not have a cell wall, so it uses these, microtube things as a structure, almost like the endoskeleton for us. Here we have our prokaryotic cell, and I probably should have mentioned this earlier, when I was talking about the prokaryotic cell, but its ribosomes secrete enzymes outside of the cell, which then digest food, which the cell then absorbs into itself. So the prokaryote digests food externally. Kind of a weird concept for us, but that's how it does it. So then at some point, the first step in the prokaryotic evolution, the cell wall would have had to dissolve, dissipate, fall apart, whatever you want to call it. It would have left. No more cell wall. This would mean the prokaryotic cell lost its structure, and it would become a lot more bumpy, like this, which would have increased its surface area. And with an increase in surface area, that means it has more area to secrete enzymes and then absorb food, so it can get much bigger. And that solves the problem as to why the eukaryotic cell is so much larger than a prokaryotic cell. Now, I should mention that at this point, it is easier for the eukaryotic cell, or prokaryotic cell, however you want to look at it, to get bigger instead of multiply into several new cells. So, since it's easier, it would do that. So, that's why it got bigger instead of multiplying into cells, in case you were wondering. Now, at this point, the large prokaryotic cell, or unevolved eukaryotic cell, however you want to look at it, parts of the cell membrane would have broken off and then kind of journeyed toward the center and then fused together, forming the nucleus. So that's how the nucleus was created. And then other parts of the eukaryotic cell would have broken off and, formed the Golgi apparatus, which is pretty much an oddly folded structure made out of cell membrane that packages protein molecules. And then we'd also see the endoplasmic reticulum form at this point, which is pretty much just a long pathway, which would also be formed from parts of the cell membrane breaking off. So here we have our fairly evolved prokaryotic cell. And remember, it digests externally by secreting enzymes. And this area here is a very efficient method, this kind of slit thing, because there are a lot of enzymes in here, which food would then go in, and be digested quickly, and then absorbed. And by natural selection, an efficient method becomes more common. So this becomes kind of common, so food will go in here and then be digested. Now, a natural thing of this type of cell is to pinch parts of itself off. So, say these two get a little closer, and then that black mark is not really working really well, but it would pinch this off then. So this would be pinched off, and it would kind of form like a bubble almost. So it would then kind of form a bubble, which would have a lot of digestive enzymes in there, with also some food. But this would pretty much be the lysome. So that's how it would work. It would be these kind of slits, which would then be pinched off with a bunch of digestive enzymes in it, which would then be used to digest food. So that is how the development of lysomes came into the prokaryotic cell. So here we have our slightly unevolved eukaryotic cell. Now, the really last thing missing is mitochondria. And how do we get those? Well, mitochondria are actually very similar to prokaryotic cells. So, what we understand to have happened is we have this prokaryotic cell here, which this cell, this larger cell, would have absorbed and kind of, eaten, essentially. And it would have been in there. Now, on rare occasions, the food won't be digested and it will be just absorbed. So, at some point in the history of these cells, which is, a couple billion years, I think, this would have likely happened. This mitochondria would have been absorbed into the cell without being digested. And that is how mitochondria got into it. They would have been prokaryotic cells that were absorbed. And the DNA that was in here would have left there and gone into the nucleus, making that even more DNA, and then officially becoming part of the eukaryotic cell. So here we have our eukaryotic cell. And I should point out, there are more than one of each of those organelles. I just drew one to keep it simple. And there are actually other organelles in eukaryotic cell. I just don't know what they look like, and they're not as popular, so I didn't draw them. But there are other organelles in that cell. And they would have been absorbed in a similar way to mitochondria here, like chloroplasts in plant cells. They would have been absorbed in a similar way as the mitochondria here. And now we have something called the oxygen holocaust. Because initially on Earth there was no oxygen. Oxygen is a product of organic life. Because chloroplasts in plant cells take sunlight and the hydrogen molecules from water as energy. And this just leaves oxygen molecules free to float around, which are actually poisonous to eukaryotic cells, assuming they don't have the mitochondria. Because they produce, like, hydrogen peroxide, which isn't good for the cell. So, what these cells had is a thing called peroxomes. They don't use oxygen as a form of energy or anything. They just convert it into something that won't kill the cell. So, they just exist to keep the cell alive by not poisoning it with oxygen. Other cells that didn't have these would have died out, kind of eliminating a lot of the links between prokaryotic cells and eukaryotic cells, and some prokaryotic cells would have been able to hide in oxygen-free environments. And then at some point, because the mitochondria uses oxygen to actually create energy, which is, more efficient and better, they would have been involved, and not just, considered useless. And peroxisomes, which now become useless in terms of oxygen converting, also provide other uses in the cell, so they would not have been just kicked out. Well, hope you enjoyed the video, and see ya!